Good day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Dita Romagwala, and this program is brought to you by the Northern Cape Department of Education in collaboration with Pagama Research and Development. So today we are looking at our grade 12 examination revision. We are still focusing on DNA, the code of life, and we will be looking at the process of DNA replication as well as DNA profiling. So here we're just going to look at ways in which we can better answer questions on DNA replication, as well as questions on DNA profiling, and also how to interpret DNA profiles. So let's just have a look at the topic content that we have for these two topics. So we have the process of DNA replication. So what you must know here is when in the cell does DNA replication take place. So you must know when it takes place. You must also know where it takes place, right? And then you must know how DNA replication takes place. It means that you must be able to describe the process step by step, which is one of the main things, because for this question, you're either going to be given a diagram to label, or you're going to be requested to describe the process for about 6 to 7 marks. So very important for you to simply just know how to describe this process, and you will be able to score those marks if you are asked a question on that. You should also know the significance of DNA replication. So why is DNA replication important? Why do we is it important for the cells to undergo DNA replication, or for DNA to undergo DNA replication? And then when we're looking at DNA profiling, interpretation of the DNA profiles. So first, you must also know what is the difference between a DNA profile and what is DNA profiling. Whereas DNA profiling is the technique, whereas a DNA profile is the black bars or the black bands that we are going to be looking at and interpreting. Okay, and then the uses of DNA profiles. What do we use DNA profiles for? We're going to look at the biological evidence that is used in DNA profiling. We're going to look at how it can be used in paternity testing, how it can be used in identifying dead bodies, and other uses. All right, okay, now our examination paper. It is very important for you to understand what your examination question paper looks like and what kind of questions you will be coming across in your exam paper, right? So firstly, this is a 2.5 hour paper, right? It means that it is 150 minutes paper. Now, in life sciences, 1 minute is worth 1 mark. It means that if you are writing out of 150 marks, you are going to be writing for 150 minutes. That means that you must use your time very wisely. Make sure that you answer your questions as best as you can, but you must also make sure that you do not answer more than you should. Make sure that you check the mark allocation for each question that you are asked, so that if it is 2 marks, you give answers that are worth 2 marks, and you don't go beyond that, which would be then a waste of your 150 minutes, right? If it is 5 marks, make sure that you give 5 marks, 5 points, so that you can get your full marks, instead of just giving quick answers that will not reward you with the full marks, right? Okay, now we have 2 sections, section A and section B. If you go through some past papers, you will see that it used to be section A, B, and section C. We no longer have section C, we only have section A and B. In section A, we have 1 question, 50 marks, that is question 1. Question 1 is divided into about 4 or 5 questions. The first one would be 1.1, which is multiple choice questions, right? Second one is 1.2, which is the biological terms or terminologies. And then the third one would be the column statements and matching items, right? That is question 1.3. Then we would have question 1.4 and 1.5, whereby you're going to be asked short questions. It's usually a diagram that you are supposed to label and answer questions based on the labels that you've given. So you would be asked questions like, give the letter and name of the part that is responsible for a certain function, and then you would give those letters and the names based on the question, right? That would be question 1.5 and 1.4, which will then give us the 50 marks. Section B, now here we are going to have a variety of questions, which are usually longer questions. We have your investigation questions, your calculations, drawing of graphs, drawing of diagrams, short 5-mark questions whereby you are expected to describe certain processes. So those are some of the things that you would come across here. So here we have question 2 and question 3. Question 2 is 50 marks. Question 3 is another 50 marks. That is the 100 marks that we have. So a combination of these two would then give us this 150 marks. All right, so by the end of our lesson, you should be able to describe DNA replication, describe the process of DNA replication. You should also be able to interpret a DNA profile, as well as describe the uses of a DNA profile, as we've mentioned in the previous slide. All right, now as you are preparing, just come back and have a look at these examination tips. These first 2 slides are not for any specific topic. These are the general tips that you must look at as you are going through your examination. However, in the next slides, we will be looking at the tips specifically based on today's lesson, right, of DNA replication and DNA profiling. So here we have some of the marking principles that the markers look at when they mark the exam scripts, meaning that it is also important for you to understand all of these so that as they are marking, you are complying with all of these, right? So if more information than marks is allocated is given, you should stop marking when the maximum marks are reached. That's why I was saying check the marks, make sure that you do not give more points than you are requested, because that would simply be a waste of your time. You will not get any extra marks. If, for example, 3 reasons are required and 5 are given, mark only the first 3. Only the first 3, which means that even if your answer 1, 2 are incorrect, 3, 4, 5 are correct, we are going to mark 1, 2, 3, incorrectly the first 2, correctly the number 3, and then we will leave out the last 2, because we are only marking the first 3. So make sure that you only give what you are sure is correct. If whole process is given when only part of it is required, read all and credit relevant part. This is a good thing. However, it could be a waste of time for you by giving long processes, whereas only a small bit of the question is required. Make sure that you answer as you are asked. If comparisons are asked for and descriptions are given, accept if differences and similarities are clear. I think that is straightforward. Okay. If tabulation is required, but paragraphs are given, candidate will lose marks for tabulation. So you only lose 1 mark for tabulating, which you shouldn't, because it's simply just a mark for drawing a table. Make sure that whenever it says tabulate, you tabulate. If diagrams are given with annotations when descriptions are required, candidates will lose marks. Make sure you do not do this. If they say give a diagram, if they say give a description, not a diagram. Describe in words, not in a form of a diagram. Number 7, if flowcharts are given instead of descriptions, candidates will lose marks. Also here, if they say describe in words, do not give a flow diagram. Make sure that you read the question. Make sure that the information that you are given, even before you go to, if it's question 1, even before you go to 1.1, make sure you read all of that information you are given. Make sure that you understand everything. Make sure that before you get to your question, you understand everything. And also that question, read it twice if you have to, so that you answer what you are asked. Make sure that you do not lose marks on things that you understood, but you didn't read questions properly. Okay. Caption. All illustrations. When we're talking about all illustrations here, we are referring to diagrams, we are referring to graphs, we are referring to tables. Everything, make sure that they have a heading or a caption. Simple. If you are expected to draw the structure of a chromosome, just say structure of a chromosome. Underline it and draw your chromosome. You will get a mark for that. And make sure that you do not draw diagrams that are not labeled. You would never be expected to draw a diagram and you are not expected to label it. Even if it doesn't say draw a labeled diagram, you will not lose marks for labeling it. Make sure you label, because usually you get a mark for drawing the correct diagram, you get a mark for having a heading, and you get a mark for a few labels. Usually it's 4 marks, 5 marks, 1 for the heading, 1 for the correct diagram, 3 for the correct labels. So make sure that you label your diagrams. If only the letter is asked for, but only the name is given and vice versa, candidates will lose marks. As much as we can see that you know the name, we want to know that you can identify it on the diagram. So if they say label it, do not give the name. If they say give the letter and not the name. All right, so now in this lesson that we're looking at, we're going to come across investigation questions, which I know can be a bit difficult for a lot of us, right? So I'm just going to give you a revision of how to go through these investigation questions, right? Things like your hypothesis, your variables, and validity and reliability. Let's have a look at hypothesis to start with. It says here, this is a proposed explanation made on the basis of limited evidence as a starting point for further investigation. So you give a hypothesis with limited evidence so that you can start doing further investigations, meaning that by the time you give your hypothesis, you do not have any results. You have not started with your investigation. You're still going to do more investigations. A hypothesis is given before the results of the investigation are obtained. Therefore, it does not have to be correct. Now, if I'm saying it does not have to be correct, I don't mean that you should now give a hypothesis that is not relevant to the question that you are asked. For example, if we are looking at body temperature, it says here, as the body temperature increases, enzyme activity decreases. Now, this is a hypothesis that you should give if the investigation is investigating the effect of body temperature on the enzyme activity. If it is not investigating that and you talk about this, that would be incorrect. But if it is talking specifically about body temperature and enzyme activity, this would be a correct hypothesis. Now, you are saying temperature increases, enzyme activity decreases. That's correct. Another one can say, as the body temperature increases, the enzyme activity increases. Remember, you do not know as yet what the body temperature would do to the enzyme activity. Therefore, even if you say it causes enzyme activity to increase or it causes enzyme activity to decrease, that is your hypothesis. That is what you are saying it would happen. We are still going to do the investigation, further investigation, to see if your hypothesis is indeed correct or not. Or you can say an increase in body temperature has no effect on enzyme activity. So you're saying when the body temperature increases, the enzyme activity does not change. That is also correct because that is what you think will happen. Now, this hypothesis must include both variables. Where do you usually get your variables? As you read the information that you're provided with, you would be able to pick out that this is my independent variable, this is my dependent variable, which now takes us to the second part. We're looking at variables. We have 3 variables: independent, dependent, and the controlled variable. Now, with the independent variable, this is the factor in an experiment that the researcher intentionally manipulates or changes to observe its effects. So the investigator would keep changing this independent variable so that they can see what happens to the dependent variable when the independent variable is changed. Right. So, for example, we cannot really change the body temperature, but let's say we are changing the room temperature, right? So, what would happen if we change the room temperature? We switch on the heater or we switch on the aircon, depending on whether we want it to cool or hot. What happens when we change that room temperature? The room temperature would now be our independent variable. Why? Because the researcher can intentionally manipulate or change that temperature. Now, this room temperature, what is it going to do to something else? That something else would now be our dependent variable, because changes in the room temperature would lead to changes in our dependent variable. The dependent variable depends on the independent variable. Any changes in the dependent variable are hypothesized to be directly caused by the changes in the independent variable. True. Now, the dependent variable. This is what the researcher measures. This would be measured as it changes due to the independent variable to see if it changes as a result of the manipulation of the independent variable. It represents the outcome in an experiment. Yes, this is the result, the outcome. What happens as a result of the changes in the independent variable. Changes in the dependent variable are considered to be dependent on the independent variable. That is also true. Okay, lastly, we have the controlled variable. We hardly come across questions like this, but I've seen that these are some questions that we come across a bit more recently. So let's have a look at that. This is a factor in an investigation or experiment that remains constant to ensure that the effect of the independent variable on the dependent variable are isolated. What do we mean by isolated? We mean that we want to make sure that as the independent variable changes, it is going to lead to changes in the dependent variable, and that will be the only thing that causes changes in the dependent variable. We don't want any other factor around us to cause changes to the dependent variable, but we want the independent variable to be the only reason why the dependent variable changes. Let's have a look at an example here. Now, we have 3 flowers, A, B, and C, right? At A, we have Coke, which is our independent variable. At B, we have juice. Again, this is our independent variable. At C, we have water. It is our independent variable. Again, 3 different independent variables. So let's say we are doing an investigation. We want to check how watering a plant with these 3 liquids would have an effect on the plant. Now, that means the plant or the growth of the plant is our dependent variable. In all these pots, the growth of the plant is our dependent variable. The growth of the plant depends on the type of liquid that you pour into the plant. Right. Then the control. The control would be the pot that the plant is growing in. You can see that the pot has remained unchanged. We've used the same pot throughout, so that the type of pot being used is not one of the factors that is going to affect the growth, right? The only thing that is changing here is the type of water or the type of liquid that you water the plant with. Everything else remains the same. And then we're going to observe the changes in the growth of the plant. As we are observing the changes in the growth of the plant, we say the growth of the plant is the dependent variable because it depends on the water or the liquid. All right, moving on. You must always learn all terminologies for every topic. Even with DNA replication, even with DNA profiling, we have terminologies. Make sure that as you prepare for every topic, the terminologies are the first thing you go through, so that as you're going through your work, you know exactly what they are referring to when they use certain terminologies. Make sure you know that. This will also be useful when you answer question 1.2 in your exam, which is based on terminologies only. So you can score about 10 marks of terminologies. Eliminate the incorrect options when answering multiple choice questions. It will become very easy for you to choose the correct answer. That is also something that you do when you answer multiple choice questions. Always use a ruler when comparing the black bands or the black bars on a DNA profile. When you look at DNA profiles, you should use a ruler so that as you're comparing the band of the 2 individuals or the 2 samples, you should be able to know exactly whether they are matching or not. By using a ruler, you will not assume that they are matching. You would be able to see that these 2 are exactly aligned with each other. So the ruler makes it very easy for you to identify the matching bands. Okay, the process of DNA replication does not change. It means that once you know it, you know it. Nothing will change. If you are asked to describe DNA replication, nothing about that will change. It will remain the same. So learn it. I mean, you know exactly what you'll be asked about DNA replication. Simply learn that process. And that is it for you. You are ready. Just learn how to describe the process, which we are going to look at. And then DNA replication. In DNA replication, we have both strands that act as a template, unlike in transcription. So that is one of the things that might be a bit problematic when you look at DNA replication and you look at transcription of protein synthesis. Remember, in DNA replication, both strands serve as a template, whereas in transcription, one strand serves as a template. Okay, speaking of terminologies, let's have a look at our terminologies for today's topic. DNA replication, this is a process by which a DNA molecule replicates to produce an exact copy of itself. Cell cycle, this is a series of events that occur during the lifespan of a cell. A template, that is a strand of DNA upon which another strand of DNA is built during replication. Okay, as I said, both of the DNA strands serve as templates. A DNA profile, this is black bars that are left behind on X-ray film when an extract of DNA undergoes biochemical process. We're going to look at DNA profiles. DNA profiling, that is a process or a technique of identifying an individual by comparing his DNA with that of another known individual. So be able to differentiate between DNA profile and DNA profiling. DNA profile, we look at the black bars that are left on X-ray film, whereas DNA profiling, we're looking at the process or the technique whereby we use these DNA profiles. Now let's get into our lesson. Right, so remember we are looking at DNA replication and DNA profiling. Now for replication, let's look at the definition. This is the process by which DNA makes an exact copy of itself. It's called DNA replication. Right, so here we're going to have one DNA molecule. This DNA molecule is going to undergo the process of replication, whereby at the end of the process, we're going to now end up with two DNA molecules that are identical to each other, meaning that everything about the two DNA molecules is supposed to be the same. Okay, where does this process take place? Right, remember when we looked at our exam guideline, it said when does it take place and where does it take place? Right, so DNA replication occurs before cell division begins, right? And this means that it occurs at the start of both mitosis and meiosis. Remember, before mitosis and meiosis, we have interphase, right, in the nucleus. This happens in the nucleus. We have interphase. That is when it occurs. It occurs during interphase so that it can prepare the cell for mitosis or meiosis. Okay, so takes place in the nucleus of the cell. That is where we find our DNA. So definitely that is where we can expect this process to take place. This is going to take place whereby this DNA molecule is going to replicate itself, and then at the end, we are now going to end up with these two DNA molecules, right? Remember, the two DNA molecules are supposed to be exactly the same. This is carried out in preparation for cell division. By cell division, we mean mitosis and meiosis. All right. Now, during this process, as we've said, the DNA will make an exact copy of itself. At the end of the process, we're going to end up with two DNA molecules. Each DNA molecule will have one old strand and one new strand. Now, the old strand, as you can see here, is blue, meaning that this part here would be our old strand. Also here, we have our old strand. And then the part in red will be our new strand. This new strand is formed using the template. The old strand is our template, using the old strand as our template so that it can copy itself from there. Just like when you make copies on the photocopying machine, you place a paper, that paper serves as your template because you expect whatever copy to come out of that photocopying machine to look exactly like this original copy that you have, right? At the end, you have one original copy and one new copy, right? Okay, which is what we are seeing here. So we have our two DNA molecules. When we go further, you will see that these two DNA molecules are going to look exactly the same. Okay, why is this process important, right? You would wonder why we have placed mitosis here. This is because it's very important for the process of mitosis. As we have said, it is done in preparation for mitosis and meiosis. So the reason why DNA replication takes place is to make sure that the daughter cells, the cells that are produced at the end of mitosis, right? The daughter cells that result from mitosis would each contain identical genetic material. Now, we have our DNA, right? This DNA then replicates, and then we have two strands that are now the same. These two strands are then going to be shared amongst the daughter cells at the end of mitosis. Now, remember, if this process had not taken place, it means that mitosis would start with this cell. If it starts with this cell, how were the two cells going to share the genetic material? However, here, because we have this DNA strand, this DNA molecule with the identical strands, they are going to be easily shared amongst the daughter cells. Okay, so each cell will contain identical genetic material, and this genetic material will be also identical to the parent cell, as we can see. Remember, mitosis results in cells that are genetically identical, meaning that they have the same number of chromosomes and the same type of chromosome as each other and the parent cell. Without DNA replication, that will not be possible. Now, this also occurs to ensure that the two new cells that are produced at the end of mitosis contain the same amount of DNA as the parent, as I've already said. Now, during meiosis, one round of DNA replication is followed by 2 rounds of cell division. Here we have meiosis 1 and meiosis 2. It results in 4 cells that are haploid. This means that they have half the number of chromosomes as the parent cell, unlike in mitosis. So now, this will make sure that those cells are shared equally as well. And we have crossing over of the DNA that would exchange the genetic material, which is why we have cells that are not the same during during meiosis as compared to mitosis. All right, now let's just quickly summarize that process. When you have a look at this diagram is very important. Make sure you check out this diagram. You can also find it in your Mind the Gap textbook. Make sure that you note the parts that have been numbered at 1. What happens? That is your first point. That is your first mark. The double helix unwinds. Right. First thing that you're going to give. Right? Secondly, what happens? The weak hydrogen bonds between the nitrogenous base break and the 2 DNA strands are going to unzip. Second point. Right. Thirdly, each DNA strand, both DNA strands serve as a template on which its complement is built. So this will serve as a template. This will also serve as a template. Remember, they've separated now. We have 2 of them separate from each other because the weak hydrogen bonds between them have been broken. Now they both serve as a template so that a complement can be built. What is a complement? It is this complementary strands. The new strands that are going to be formed are complementary to the old strands. What do we mean by complementary again? We mean that the nitrogenous bases of the old strands are going to pair with the correct nitrogenous base of the new strands, whereby we mean that adenine would pair with thymine and cytosine will pair with guanine. Let's have a look at that. Now, at 4, free DNA nucleotides from the nucleoplasm build a DNA strand onto each of the original DNA strands. The original DNA strands are our templates, right? by attaching complementary nitrogenous bases. Complementary nitrogenous bases. That is where we are forming the complements now. Then 5, 2 new DNA molecules result. Don't forget that these 2 new DNA molecules are identical to each other. Each molecule consists of one original strand. We have the original strand over here. I'm going to highlight the original strands in the color red. We have the original strand over here. And then we have another original strand over here. Right? And then we have the new strands as well. So the inner ones are going to be our new strands, as you can see there. Let's just change the color so that we can be able to see them. The ones in blue are the new strands. The ones in red are our old strands. So each DNA molecule now has a new strand and an old strand, right? You should also mention that the process is controlled by enzymes. Now, as you are looking at this, like I said, it should guide you. Point 1, you're talking about unwinding. Point 2, you're talking about weak hydrogen bonds breaking and the strands unzipping. 3, you talk about that these 2 strands are now going to serve as templates. 4, you talk about the fact that the templates are going to be used to pair with the complements. The complements here we are talking about A pairing with T, G pairing with C, with the free DNA nucleotides that are from the nucleoplasm. And then 5, we have the DNA, 2 DNA molecules that are identical to each other, and we can see that the process also is formed by or is controlled by enzymes.